Okay.
Okay. We ready? Yeah. All right. Good afternoon, everyone, and happy Earth Day.
Oh, yeah.
So I'd like to introduce Sophie here. She got her bachelor's degree at Rhodes College in Environmental Science in 2016. And after that, she did a number of internships in science communication at the Toledo Institute, bilingual studies or bilingual education at Colorado Reef Alliance or River Alliance, and most recently, educational leader in marine conservation at Reef or Reef Educational Environmental Foundation. In the MMES cohort, 2019, so yay. 2019 cohort. Sophie received a number of awards and fellowships, and while at UVI, and the most recent was International Coral Reef Symposium Student Travel Award to present her thesis this summer at Bremen, Germany. So,
yay. Wow, thanks everybody.
Let's hope the world doesn't fall apart between now and then. So I want to, coming up in the future, before I introduce her for her thesis, she was accepted in a PhD program at Florida International University with Dr. Jennifer Rihaj, and her thesis or dissertation is going to be an impact of loss of seagrasses to commercial groupers. So that's super exciting news. So without further ado, her thesis that she's going to be presenting today is evaluating the impact of invasive seagrass on juvenile yellowtail snapper. All right,
thanks,
take it away.
Alrighty, let me just make sure everything. All right. Yeah, so thank you so much. Thanks for that, Rick. I want to just give a big thank you to all of you for being here, and a special thanks to my committee members, Dr. Sinai Haptis and Dr. Damian Willett, who are tuning in on Zoom. So as Rick said, the title of my presentation today is evaluating the impact of the non-native seagrass, Halophila stipulacea, on settlement, survival, and condition factor of juvenile yellowtail snapper, Ocyurus chrysurus, bear with me, in St. Thomas, USVI. Oh, yes, thank you. Let's see. Okay, can you see it now? How do I minimize this? Like that, perfect. Okay.
Yes, looks good.
Perfect, thanks, Damien. Awesome. So before I dive right into my project, I just want to give a little bit of background about myself. As I was searching around for different graduate school programs, everybody always asked me, oh, you know, what do you want to study? And so I always knew I wanted to focus on fisheries management. However, there's obviously a number of different avenues you can go within fisheries management. So I always knew, you know, commercially important fish is something I'm interested in, but I'm also really interested in invasive species. But I don't know, I mean, juvenile fish are pretty cute too, so maybe that's what I want to study. So when Rick presented this potential project to me, I honestly could not have dreamt of like a more ideal project. And so I'm excited to share with you all what I've been working on over the last 3 years. Why isn't it working? Let's see. What, Joe? Do you know?
No,
Arrow? Okay, thank you. All right, so before we dive in, I want to give a little bit of background on the fishing industry within the US Virgin Islands. So historically and currently, it is one of the most economically important industries. It was estimated by the NOAA 2016 economic report that roughly $7.4 million are brought in annually from this industry, which equals roughly 1.4 million pounds of seafood. This is also brought in by about 215 commercial fishermen, which was estimated by Yandel et al. In 2020. With that in mind, it's important for management to maintain these populations, and through that management, it's important for them to focus on different ecosystems. However, one of the most important but often overlooked ecosystem is seagrasses. So seagrasses do provide essential nursery habitat for many juvenile fish species. These ecosystems are important not only because they support a diverse ecosystem that provides the major food source for juvenile fish, and additionally for large megafauna like sea turtles or also stingrays. However, because we're focusing on fisheries management, the blade structure of seagrass is very important during juvenile fish's vulnerable stage from predator species. One of these economically important fish species is the yellowtail snapper, as I mentioned previously. It has been estimated by Olson et al. In 2007 that yellowtail snapper was the #1 fish species brought in, which made up about 22% of all landed species within St. Thomas. Not only is this an economically important fish species here within the territory of the Virgin Islands, but additionally all throughout the Caribbean. As you can see, their range goes from the northeastern part of the United States all the way south to Brazil. However, even though they are important, they are not currently ranked by the IUCN Red List because they are considered data deficient. What we do know is that yellowtail snapper are considered opportunistic feeders, meaning that they don't feed solely at one point of the day during diurnal feeders or nocturnal feeders. We also know that as juveniles, they are primarily planktivores, but as adults, they are considered generalist carnivores, meaning that they don't really have a specific food type that they primarily feed on. We also know that as juveniles, they do depend on seagrass habitats, which is why it's important for management to better understand how these seagrass habitats could be impacting the yellowtail snapper. Within the Caribbean, we do have 4 major seagrasses. However, for the purpose of my study, I'm only going to be focusing on Syringodium filiforme and Thalassia testudinium. Syringodium filiforme, which is also locally known as manatee grass, has a long, thin blade that almost looks like a spaghetti noodle, is how I keep it in mind. Thalassia testudinium is locally known as turtle grass, and it has a long blade as well, but it looks more like a fettuccine noodle, right? I mean, you can tell from the images, right? These seagrasses are often are highly impacted by multiple anthropogenic threats, such as invasive species. Halaphala stipulaceae was first spotted off the coast of Grenada in 2002 in the southern Lesser Antilles, and then has since migrated up to St. Thomas and arrived here in 2013. Halophila stipulaceae previously had invaded the Mediterranean Sea and is known as a Lesepsian species because it migrated after the opening of the Suez Canal. Originally from the Red Sea, this species has a large adaptation within the Caribbean for salinity and temperature, which makes it a highly successful invasive species within the territory. It has been known to outcompete many of our pioneer species like Syringodium filiforme, and could be causing a large composition change within the ecosystem. Therefore, as this change occurs, it's important for management to know how that could be impacting our commercially important fish species like yellowtail snapper. Previous studies have tried to identify how Halavala might be impacting fish populations. A study by Ollinger et al., which was actually the MMES cohort study from 2015, found that a higher abundance of individuals was caught in Halophila seagrass habitats than in native Syringodium, native Thalassia, or sand habitats. This study was conducted in Frenchman's Bay, Lindbergh Bay, and Spratt Bay, which is important to keep in mind because I also sampled these bays. I'd also like to draw your attention to the findings of this study because the primary family of species that was found was actually our snapper species. Another study coming out of the MMES department that was trying to identify how the non-native Halophila stipulaceae is impacting fish was a study done by Damon Green, yes, who looked at the condition factor of juvenile Nassau grouper. Damon found, or using condition factor as a proxy for the health of juvenile Nassau grouper, which is a measurement derived from the weight and length of an individual. And it's often used by fisheries management because it's a non-lethal way that you can estimate the health of a fish. The higher the weight, it's often considered at a specific length, it's often considered to be a healthier fish because they have high fat content. Green found that individuals caught within the mixed seagrass habitats had significantly higher condition factor than individuals caught in the invasive Halophila seagrass habitats. Additionally, trends from his study suggest that the native seagrass, that individuals caught in the native seagrass also had higher condition than those caught in the non-native seagrass habitat. Which brings us to my study. So my study, I was trying to focus on commercially important fish species. And so the question arose of what are the effects of the non-native Halophila stipulacea on the demographic rates of yellowtail snapper? The primary research questions we sought to answer was, does Halavala have an effect on settlement of yellowtail snapper? Does Halavala have an effect on the survival of yellowtail snapper after settlement? And then finally, does Halavala have an effect on the condition of yellowtail snapper as well? To answer these questions, we focused our study in 5 bays around southern St. Thomas, with A representing Saba Island, B representing Brewers Bay, C representing Lindbergh Bay, D Sprat Bay, and E is Frenchman's Bay. As you can see in the lower portion of this figure, the zoomed in images give you an idea of where the seagrass habitats are located, with the red boxes representing Halavala, the yellow representing Serengodium, and the green representing Thallassia. We focus on these bays, again, not only because Olinger also sampled these, so it would be a nice comparison to see how my data compared to Olinger, but additionally because they had the presence of large monotypic strands of both native and non-native species, or seagrass species. All except for Brewers Bay. However, we selected Brewers Bay not only for its relative proximity to the university, but additionally because it has large monotypic strands of Halophila stipulacea in both shallow and deep water habitats. So that would give us an idea of how these two areas might differentiate in the different factors that we were analyzing. The first question we sought to answer is, does Halaphila have an effect on the settlement of yellowtail snapper? To do this, 8 by 8 meter plots were established in each of the different seagrass types. The 8 by 8 meter area was determined by a previous study from Watson et al. 2002 that found that the average area of a newly settled juvenile yellowtail snapper, which for the purpose of my study and their study was less than or equal to or less than 30 millimeters. And Watson et al. Found the average home range of individuals in this class size to be about 6.3 meters. So it gave us a little bit of a buffer. From May 2020 to June 2021, visual surveys were conducted 3 to 7 days prior to a new moon. Again, the new moon was determined based on the Watson findings that peak settlement of yellowtail snapper occurred a few days prior to the new moon. Within each plot, 2 divers swam parallel to one another, carrying the 2-meter T, as you can see in this photo, with markings indicating 1 and 5 centimeter size classes. And these size classes, this not only helped the diver get an accurate estimate of the size of the fish it was seeing, but additionally, as the T was pushed along the top of the seagrass, it would divert the diver's attention to the movement. Therefore, they could properly identify as well as allocate it in the correct size class to get an idea of the abundance of settlement that occurred within each plot. A pulse was determined, a settlement pulse was determined as 10 or more newly settled individuals equaling less than equaling or less than 30 millimeters, as I said. These data were determined using individuals observed in less than or equal to 30 millimeters again, and only from Saba Island, Lindbergh Bay, Sprat Bay, and Frenchman's Bay. And that is because Brewers Bay, again, only had monotypic non-native seagrass habitats. Because my data did not reach or meet ANOVA assumptions, we ran non-parametric Kruskal-Wallis and Dunn's tests. The ultimate findings of my study were that Halaphila had a significantly higher settlement than either of our native seagrasses, as you can see in this figure. Along the y-axis is the average percent of individuals observed per seagrass type, and then along the bottom demonstrates the different seagrasses. Results of this study do correlate with the findings of previous studies, such as Ollinger et al., that saw a higher abundance of yellowtail snapper in the Halaphila seagrass habitats. However, it also contributes to a better understanding on how settlement and individuals in the smaller size classes were also observed between the different seagrasses. The second question that we sought to answer with this data is when did peak settlement occur? Again, because the data did not reach ANOVA assumptions, a Kruskal-Wallis and Dunn's tests were run. Along the y-axis again is the average percent of individuals, and along the x-axis is the month of that settlement occurred. We found that peak settlement occurred in May 2020 and June 2020 as well. And then we had smaller settlement pulses in August, October, and May of 2021. A previous study out of Puerto Rico and the US Virgin Islands found by Jarovitz, 2021, found that peak spawning in adult yellowtail snapper occurred in April and May. And therefore, after spending 30 days in their larval form, it was expected that we would have higher settlement pulses in May and June. So my findings do correlate with other studies. Oh, additionally, we found that the peak settlement occurred between 0 and 4 days prior to the new moon, which also correlates with the Watson et al. Findings. We also found that Saba Island had significantly higher settlement than either of the other 4 bays. So again, we did not include Brewers Bay in this analysis because Brewers Bay only had non-native seagrass habitat. Yes, so this is important for management to potentially keep in mind as questions might arise as far as what the impacts might be on yellowtail snapper. And with Saba Island being located pretty far, as you can see in this figure, from many of the other bays, it's something that maybe should be tested further. So to answer this overall question, yes, Halavala does have an effect on settlement, and in fact, it actually significantly higher settlement occurred in Halavala than our native seagrass types. However, to dive a little bit further into this analysis, we wanted to look at a couple of environmental variables since settlement is such a complex characteristic of many fish. One environmental variable that we collected were seagrass characteristics. To do this, 10 by 10 centimeter quadrats were randomly placed among each of the different plots, where divers would record the percent cover, the blade density, or also known as the blade count, and then 5 counts of the maximum blade height. So to get percent cover, it was just looking at each of the individual quadrats and quantifying roughly how much of that specific seagrass you saw. Yeah, and then again, blade density was just blade count, and then 5 counts of maximum blade height within each quadrat. These numbers were average for each of the plots, and then that data was used to quantify the difference of these characteristics. Can y'all see that? I don't know how to get rid of this black thing. Does anyone know? Mm-hmm.
Hide floating meeting controls.
Hide. Where?
Floating meeting controls.
Floating.
Hide, just above your arrow.
Oh, wow, right in front of my face. Thank you. So what we found is that Halaphila had a significantly higher percent cover than our native seagrasses.
Hit a button on one of the remote controls. TV.
Oh, thank you. Sorry, everyone on Zoom, we're having some technical difficulties. Okay, perfect. Okay, Halaphila also had significantly higher blade density or blade count than our native seagrasses. And then additionally, Halaphila had a significantly shorter blade than our native seagrasses. Another aspect we looked at was the temperature and dissolved oxygen within each of the different bays. So to do this, 1 hobo temperature logger was placed in each bay, except in Brewers Bay, where 2 temperature loggers were placed, 1 in shallow and 1 in deep. These loggers were placed from May 2020 to September 2021, at which point they were picked up and swapped for one mini.02 logger. Only one logger was placed in Brewers Bay, and we put it in deep Brewers Bay. We placed each of these in the center of a plot on a steel rebar, as you can see in this photo. The average monthly temperature was used to better understand the variance among each of the different bays over time. There was no significant difference of temperature among the bays. However, dissolved oxygen was pretty distinct dependent on the bay. To get this data, we removed all of the outliers, which we considered 12 milligrams per liter or higher. Additionally, we collected data on prey availability per seagrass type. So one passive tube trap was placed within each of the different seagrass types. The tube trap is a 60 centimeter by 60 centimeter in length and 5 centimeter in diameter, and it was filled with 10% buffered formaldehyde and sea water, and then a little bit of food coloring that you can see in the green within the photo. This not only helped camouflage the trap a little bit, but additionally, it gave us an indicator on how much formaldehyde was within each of the different traps. The concept behind this is that anything that floats or passes over the top would fall would immediately be fixed. And we were able to estimate the species richness among the different seagrass types. These traps were placed out from May 2020 to September 2021, and they were collected on a bi-monthly basis. Once they were collected in the field, they were processed through a 75 micrometer sieve and immediately replaced within the plot. Once back in the lab, they were processed through a macro, coarse, and fine filter, which were 2 centimeters in diameter, 0.5 centimeters, and 0.075 millimeters respectively. We were able again to identify the plankton to species richness, or using at least to the ordinal level, and then we determined the species richness based off of these. Got it, Lori? Okay. We did not find a significant difference of species richness among bay or seagrass type. However, trends do demonstrate that native seagrasses could have a higher species richness, although this was not a significant difference. The only bay that we did not see these trends were Saba Island, and therefore it's recommended that further research is conducted in this area. To understand how these environmental variables might be impacting the settlement of juvenile yellowtail snapper, I ran a generalized linear model. From this model, we were able to determine that month and temperature were significant predictors for settlement using the Gaussian distribution. However, the deviance between the null model and the residual model is quite large. Therefore, my model might not be the best fit. However, we did find a reduction in deviance to about 25% that demonstrates that although it might not be the strongest fit model, it does demonstrate that these variables might have some influence on settlement. The next question we sought to answer was, does Halaphala have an effect on the survival of yellowtail snapper? To do this, on scuba, a diver would use 2 clear vinyl-sided nets to catch juvenile yellowtail snapper, and we were targeting individuals that were about 5 centimeters or less, kind of like this guy in the photo. Once they were caught, individuals were then transferred to Ziploc bags. Oh, sorry. Oh my gosh, I got way ahead of myself. I'm just eager. Okay, sorry. The second question we sought to answer was, does Halavala have an effect on the survival of yellowtail snapper? So to do this, once a settlement pulse was determined, again, that was 10 or more individuals, divers would return to that same plot where they would continue doing visual surveys daily for 7 days, then every other day for 8 days, and then weekly for a month or until the next settlement pulse was detected. After 0 individuals were recorded within each of the different plots for 2 consecutive surveys, the surveys were halted. Okay. Okay, sorry, I got a little bit. These data were used to determine the rapid mortality of individuals among the different seagrass types. So in this figure, along the y-axis is the percent persistence. And how we determine percent persistence was by taking the daily population, or a population that we saw on any given day following peak settlement. Peak settlement, again, is the highest number that we recorded within each of the plots. What we found is, the dotted line is representative of Syringodium. The solid line is representative of Thallassia, and the dashed line is representative of Halaphila. Each of the different dots represent a count for the daily persistence conducted. The overall daily mortality rate on average for about 20 days was seen as 4.65 loss within Syringodium, about 2.73% loss within Halaphila, and about 2.35% loss within Thallassia. Compared to the findings of Watson et al., who only conducted surveys within Thallassia, my overall percent persistence of the population was about 3% after 24 days. In comparison, Watson found a percent persistence of 21% after 30 days. Results from my study demonstrate a lower percent persistence within the population. So to answer this question, does Halaphila have an effect on the survival of yellowtail snapper? We don't really know, although trends do demonstrate that Halaphila and Thallassia have similar mortality rates, which are both higher than the Syringodium seagrass habitats. Okay, the third question we sought to answer now is, does Halaphila have an effect on the condition factor of yellowtail snapper? To answer this question, as I told you before, a diver would swim with 2 clear vinyl nets and catch individuals that, again, we were targeting about 5 centimeters or less. These individuals were then transferred to their own Ziploc bag underwater, and then once returning to the boat, they were placed on ice, which humanely euthanized them. Upon getting back to the lab, I would then measure them for their length as well as their weight, and so we could derive their condition factor. In total, 260 juvenile yellowtail snapper were caught, 116 from Halaphila, 66 from Syringodium, and 78 from Thallassia. Again, how we derived condition was using their total weight divided by their length cubed and then multiplied that by 100. Similar to my settlement data, condition did not meet the condition data did not meet parametric assumptions of an ANOVA, and therefore we ran a Kruskal-Wallah test. There was no significant effect of Bay detected on the condition of juvenile yellowtail snapper. However, similar to the green study, we did find that the native seagrass had trends of having a higher condition than those caught in Halaphila. If our numbers had increased, we potentially could have seen a difference, but we just don't know, and therefore more research is suggested. We did find, however, that Bay had a significant effect on condition of juvenile yellowtail snapper. We found that individuals caught in Brewers Bay Deep had significantly lower condition than those caught in Brewers Bay Shallow, Frenchman's Bay, and Sprat Bay. Additionally, we found that individuals caught in Saba Island had significantly lower condition than those caught in Sprat Bay. So to answer this question, does Halavala have an effect on the condition factor of yellowtail snapper? The findings from my study do not suggest that they do. So this is something, again, that potentially more individuals could have given us stronger results. However, it also shows promising results that it was not significantly different between the different seagrass types. Again, I ran a generalized linear model to understand which environmental variables might be having an effect on condition. Using the Gaussian distribution, I found that dissolved oxygen, temperature, and percent cover could be are significant predictor values for condition. Again, the variance was quite large between the null and my model, my predictor model that I ran. However, there was a reduction of about 10% between the two model types, which shows that although, again, it's not a great fit, there is some indication that these variables could be having an influence on condition. So I know I just threw a bunch of results at you. So a little bit of some key takeaways from my study again are that higher settlement was detected in Halavala habitats. Similar survival rates of juvenile yellowtail snapper were observed between Halophila and Thesis seagrass habitats, which were higher than survival rates found within Serengodium habitats. And then thirdly, there was no significant difference of yellowtail snapper condition factor among the different seagrass habitats. So this brings us all to the broad overarching question that I know everyone is asking themselves, which is what are the management implications of my study? Oftentimes when I discuss my study, one of the primary questions I'm asked is, based on the results of my study, do I consider Halavala a non-native or an invasive seagrass? Personally, from the results of my study, it seems that Halavala might just be a non-native because it's not having a negative implication on yellowtail snapper populations. And I would like to take Brewers Bay as a bit of a case study to explain why I feel this way. So again, within Brewers Bay, we had a plot in both shallow and deep water habitats, which you can see here on the map. The shallow is closer to the shoreline and the deep was a little bit further out. These plots were located in 3 meters and 16 meters respectively. We did not find any significant difference of settlement among the two plots. But however, again, we did find a significant difference of condition factor from individuals within the two locations. The reason that that Brewers Bay is a good idea of how Halavala is affecting yellowtail snapper is because these deep water habitats previously, it's far beyond the range of native seagrasses, and therefore we can assume that before the presence of Halavala, these areas were most likely only bare sea floor. So the fact that any settlement occurred within this area is a promising result. And as you can see from these data, yellowtail snapper had higher settlement in August and October in the deeper habitat than in the shallow habitat. So this indicates that yellowtail snapper are taking advantage of this deep water habitat, which again is promising as Halavala might be expanding their area and potential habitat space for settlement. This additional suitable settlement habitat could be beneficial for their populations. However, more research is necessary to determine in the next size class how these deep water habitats could be affecting their populations. So to go back to the overall question, what are the implications for management of my study? Again, I want to reiterate that prior to us making a conclusion on the impact of Halavala among the entire local ecosystem, we need to answer even more questions than what my study does. So my study was able to fill in some of the gaps that were previously asked about how Halavala is impacting the survival, as well as the settlement rates and the condition of commercially important fish species, as well as we got some understanding of how these deep water seagrass habitats could be influencing the settlement of individuals. However, as you can see, the concept behind the food energy water system is a complex understanding of how all the variables within an ecosystem are interacting with one another. So my study really focuses more on the food portion of the fuse system. However, in order for management to get a broader understanding of the impacts of Halavala, they should focus on some different areas. So some suggested, but not limited to, a couple of future studies that I believe would provide more information on the impacts of Halavala would be to focus on the water quality, and potentially if the water quality is different between the native and non-native seagrass habitats. And then additionally, to look specifically at herbivorous fish that rely solely on seagrass habitats as a major food source. For instance, the bucktooth parrotfish, which I did see just, anecdotally, eating on both native and non-native seagrass habitats. So that might be a good fish to analyze further as far as the condition of that specific species to determine the broader implications of Halavala. However, nonetheless, my results do provide a bit of a silver lining as far as the impacts of the non-native Halavala stipulacea species. Again, we found that higher settlement rates in the non-native seagrass, which do provide promising information to management, and could demonstrate that many commercially important fish might also be utilizing this habitat. However, more information and research is needed. So I would like to say a large thank you to the generous funders of this project, including the NOAA Coral Reef Conservation Program, the National Science Foundation Strong Coast Program. Additionally, I'd like to thank the MMES department and the University of the Virgin Islands, and finally the Lana Vinto Charitable Trust for their generous support of this project. Secondly, there are so many people that I have to thank for your love and support. All my friends and families and colleagues, you all know who you are, that have offered so much support throughout this journey. I honestly could not have done this without all of you, so I greatly appreciate it. And do I have any questions? Yes, Joe.
So I have a kind of question about, you were talking about how important these are for juvenile browns. How long are these yellowtail snappers kind of making use of this area?
That's a great question. So the Watson study found that yellowtail snapper depend on seagrass habitats until about 7.5 centimeters, at which point they experience an ontogenetic shift out to the ecosystem. So she didn't look at age necessarily, but about 7.5 centimeters is when they transition out. Lori, did you have a question?
Fascinating.
Thank you.
I knew a little bit about it until now, but this is great.
Thank you.
Excellent presentation.
Thank you,
Way
Lori.
to manage the technological challenge.
That was, yeah, COVID, right?
That's right. Teaches us all these things. So I guess my first question would be, I guess it's your third study, your third question methods.
Yes.
That's your third question. Did you give any thought to catch per unit effort? I just noticed a big difference.
No, we did.
Well, you thought about it. I just saw it.
Yes, we thought about it, but the way we caught fish was basically, as efficient as possible, because as I was out, or divers were out collecting the settlement surveys, another diver would go along and collect yellowtail snapper. However, they were located we try to focus about a meter outside of the plot. So not necessarily catch per unit effort, but hopefully the data that we collected from the surveys gives us an idea as far as like the broader population.
So you are saying, my understanding is of what you just said is that you are saying that you gave approximately the same effort to each site.
Yes,
Each type of seed.
yes, to each
Not
type
really
of
the
seed.
same.
Yes, exactly.
Second question, follow up,
Yes,
has to do with how different these different seagrass meadows.
yes,
And so, and, we all have in our mind some particular seagrass meadow. We can mention one.
of course.
I have this one favorite, Thalassia. And I gotta tell you, that method with the tea wouldn't work. But I'll bet you your Thalassia were more like that last image before this one.
Which one is?
I'm just wondering if
Oh.
your Thalassia were more like that height. I didn't quite remember what your height was when the question hit my mind. You had measured the height, maximum height of these different ones. What was your height on, if you go back to that?
Yeah, of course.
Because, Thalassia under sparse, very different habitat.
Right. Actually, it might be better if I just escape
And I was
this.
just thinking, we're going forward, sorry, I can't tell you what. Somebody followed up with the new T, right? With Rotenone, just to see how it compared. Because we're trying to do away from Rotenone. And I don't, well, that Thalassia didn't compare too well.
Oh, interesting.
Because Thalassia has this robustness to it, especially if it's tall. So that's the height.
So we found on average about 12 centimeters in length.
Yeah, so I see that. So you got your-
However, we did see, so Yellowtail Snapper, I didn't collect information on their behavior. However, in the smaller size classes that we were analyzing, they are extremely site dependent. So as we would arrive to a plot, and again, this is only based off my own personal observations, what we would see is they would be hovering above the seagrass. And so we were able to get an idea of how many individuals were present. And it honestly, they wouldn't dive down. They were almost extremely naive. They wouldn't dive down until you were almost right on top of them with the PVCT.
You heard me count them before you got it.
Exactly. Yeah, exactly. So the PVCT really did help with the larger size classes. But yeah, as far as like the newly settled individuals, they really, I mean, their behavior was so unique. And it was really interesting because I would have thought the same as what you're saying, that they were hiding within the seagrass blades, but they weren't really.
And for species,
Right.
we weren't talking about all these fishes that live in this.
Right, exactly.
And it does.
It does.
That's what you said.
Yeah.
I love that you know that.
I love them.
My last question.
Sorry, what was that? Oh, that's Sam Giddings. Yes, he's amazing. I think he's on Zoom. Yeah, he helped our lab out last fall. So yeah.
Thank you so much.
Of course. Yes, Kayla.
My question is, like, from the fisheries management perspective, Halaphala has been around, like, I guess we said since 2013, right?
Mhm.
Kind of boomed after hurricanes in 2017. So that's,
Right.
like, at least 5 years that potentially Yellowtail Snapper getting this, like, movement or this settlement first,
Mhm.
potential new habitats for it to settle. And I'm wondering if that is coming out in the landings data at all. Like, what is people landing Yellowtail Snapper? Is there been increases since the?
I honestly don't know. Yes. So the question was, since Halavala has been around roughly since 2013, is it affecting the landings data reported by fishermen? I personally can't say. I would have loved to have had time to conduct some fishermen surveys to see if they have seen any difference. But I can't answer that question just because I didn't specifically focus on it. I will try to find that out for you though and let you know. Yes.
You said that you left out Brewers from
Yes.
Could you just repeat why you left it out?
Yeah. So I left Brewers Bay. Oh yes. So the question was why I left out Brewers Bay in the analysis from the beginning of the study. The reason I left it out is because there was only non-native seagrass habitat found within that bay, and therefore we couldn't. It would throw off the statistics because we would have had more sampling done within Halaphala. So we removed that for the primary analysis in order to keep the consistency within the statistics. Yeah, Joe.
Sorry, last one I spoke.
No, it's fine.
But so was I hearing you right that you saw a really high abundance of settlement at Saba? You also saw a lower body condition?
Yes.
So is there an interaction between those two? And you see that?
Okay, so the question was, we saw a higher settlement at Saba, but also a lower condition, correct?
Yes.
Okay. I can't say for certain because I didn't run data on the two combined together. However, if we go back to the plankton data that we found, which is here, I would hypothesize that part of the reason the condition is lower at Saba is potentially because we're seeing a slight, like a difference in the prey availability at Saba than we are in the other bays. However, further research would be necessary. In my generalized linear model, I did not see that plankton, like the plankton trap data had a significant effect or was a significant predictor on condition. However, it is something that we could look further into. Yes.
Question, when you say that you added food coloring to the trap?
Yes.
Sorry, I missed it.
No, that's fine. So the reason that the question was, why did we add food coloring to the trap? And so the reason behind that was the formaldehyde is much heavier and denser than our seawater. And so by adding the food coloring, it gave us an idea of how much formaldehyde was within each trap. And so if there was any leakage from the bottom, we were able to quickly identify it and then replace that trap so we didn't miss any sample time. Yeah. Do you have a question? Yeah. Okay.
So it was interesting that you got more settlement in deep brewers in August and October. Can you maybe hypothesize why that would be happening at that time?
So as far as settlement, the largest indicator was temperature. And so temperature might be the biggest indicator for why settlement might be occurring during those months. There's also so much unknown about why, oh, sorry, I don't know if everyone on Zoom heard, but the question was, what is my hypothesis as to why more fish are settling in deep brewers in August and October rather than the other months? And so now I lost my train of thought. So it could be that there was a thermocline shift, or additionally, there's so much unknown as far as like why fish are actively selecting the habitats that they are. It could be that there was more prey available. I really don't know too much as to why they would be selecting during those months specifically, but further analysis would be necessary. Okay. Any other questions? Do we have any questions on Zoom?
Great presentation.
Oh, everyone's so great. Okay. Yes, Andrew.
So I have recently had the privilege to come out and try to catch fish in this lab. Can you just explain like how you would catch a fish that's just like 2 centimeters to that's
Yeah. Okay. So the question was.
in an open environment like that. That sounds really challenging.
So the question was why basically how we caught the fish, because Andrew's had a terrible time with the Newid lab so far. So yellowtail snapper at this size are fairly naive. I also had the blessing of Kayla Budd, who is that what you said? Oh yeah, who are actual professional. I mean, I swear, like if you need to catch a fish, call them, especially in this size class. It takes a lot of patience, and I think the clear, oh my gosh, I think that the clear vinyl, they couldn't see it, and so sometimes, I mean, you would definitely have to chase them, and then I had the mentality like I'm not going to give up, and then it would just be like this terrible experience for me. But I don't know, Kayla got the technique down, and basically you just take the 2 nets and you sort of like guide one in front and then guide it into that net. And then Rick came up with the great idea that like as soon as it's in the net, you have to like scoop it really quickly, because then the fish will try to like swim back down. Sorry, I'm not giving you any credit. I'm sure it's hurting. So yeah, so Rick came up with that technique and it works very well. And so yeah.
you don't use squirt guns anymore?
Use what?
Oh, these were pretty effective with them.
For what type?
For small fish. You're talking about? You're talking about
Yeah.
water.
Oh, no.
It's like a reverse spear gun. So when you pull the trigger, it sucks
Oh,
a
wow.
chunk of water into the tube.
Yeah, huh, okay. That sounds like a great idea.
We use that, I know, it's like-
I've never used that, so that would have been fun to learn. Yeah, that would have been fun to learn. Yes, Sean,
Sophie, I'm not, great talk, by the way. I'm not,
thank you.
Gonna ask you a question. I just wanted to make a comment real quick. And I think that I agree with your opinion that Halaphala is a non-native versus invasive species. And not only have I seen, in deeper water, where
Mhm.
grasses can't
Right.
grow, the Halaphala now growing, perhaps, well, obviously, providing some juvenile habitat, but also in shallow water that has become too polluted, turbid, whatever,
Mhm.
for other grasses to grow. And I'm thinking like in Inner Bay,
Right,
that used to be a very nasty mud.
right,
It's
like in
now
Lindbergh,
covered in Halaphala.
right.
And so it's nice that you're finding that there is recruitment and there is.
Absolutely.
use, like,
Well, and what was really interesting, I didn't present on this data, but we collected abundance of other fish. And it was interesting because we would get settlement pulses in the deeper Brewers habitat of fish that we wouldn't see in the shallow habitat. So fish, I mean, we would have, you remember, I mean, so many bridal gobies. We had pulses of tobacco fish. There's a little reef not far from that seagrass bed. And so it was really interesting also to see the diversity shift of fish that we're seeing in the deep water habitats that we don't see in the shallow seagrass habitats. So that would definitely be a very interesting future study, is comparing deep water habitat seagrass Halavala beds to shallower.
Time for one more question on Zoom.
Any on Zoom? I don't see any.
Okay, well, thank you very much. Everyone, now it's time for Sophie to meet privately with her committee. So if I could ask audience to leave, thank you so much for coming and taking the time to be in person.
All of them.
Great job, Sophie.
Yes, what kind of drink do I want? I did all of them.
Bye,
Okay.
Sophie. Great job.
Bye. Thanks, Jason.
We have all your-
In the park, yeah.